He is a biological engineering staff scientist and in PBD. He's the JBay Feedstocks Research Division Director of Systems Biology.
That's true,
Dr. Heazlewood received his PhD in the area of plant molecular biology from La Trobe University in Australia in 2000. And from 2001 and 3, he was a postdoc fellow at the University of Western Australia, where he used proteomics to investigate plant mitochondria.
Yeah, that's true. I really want to read this out.
Me too.
This is interesting. This is nice to hear the background. So he was awarded a UWA postdoctoral fellowship in 2004 and an Australian Research Council postdoctoral fellowship in 2005 to expand his proteomic work into analysis of protein modifications and signaling pathways in plant mitochondria using
phosphoproteomic techniques.
Thank you. He's the recipient of a number of awards and other fellowships, and using advanced mass spectrometry techniques to further investigate protein modifications in plant mitochondria. He was appointed to LBL in 2008 at JBay to investigate biosynthesis and structure of plant cell walls. I've enjoyed working with Josh when I was first down at JBay, and I'm looking forward to hearing him talk. Please welcome Dr. Josh Heazlewood.
Okay, so yeah, I guess I will talk about some aspects. So I'm going to sort of try to take it as slow as possible at the start, and then do a bit of some of the work that we've been doing at JBay, just to make it a bit easier, because I realise that the audience has probably has a bit of a biology background. So when I posed the question, where are all the proteins, I sort of realised that I'm actually really going to have to discuss what a protein is and what they actually do in the context of cellular metabolism and plant cell wall biosynthesis. So I was thinking all the way back to high school even, right? So this concept of what is DNA, right? And people always use this analogy of the cookbook, right, or the recipe book. So the DNA is in each cell, and it's like the recipe book, and it's got the instructions to make a cake. I mean, the interesting thing about this analogy is that it's sort of true, but in fact, the egg beater and all that, and all the equipment that you'd actually use to make the cake is also sort of coded for by that recipe book. So although the analogy is apt, it's slightly misleading in some regards. And it's really the egg beater and all of those components, which are sort of the proteins in the analogy of this sort of baking a cake analogy. And so the proteins are the things that do the work to actually make the cake, and also part of the cake as well. So that's sort of the somewhat slow start in terms of what is a protein and why are we interested in them, I guess. So what are proteins? So these are two sort of cells. So this is a bacterial cell. So you would have seen Adam talk about microbial communities and microbes and things like this. So when we think of a microbial cell, we tend to have a pretty simplistic idea of the structure of a microbial cell. And this is sort of just to show, supposed to sort of show a tissue, an amalgam of cells. So this is the sort of thing you'd find in a human. And really, again, back to this thing. So I'm assuming most of you have heard about DNA, right? So you have all the copies of DNA in each of your cells, and it's this sort of these sort of coding sequences that are used to make the protein. So the information on how to make the protein is in there. And so proteins. So also, everyone just jump in with a question or something like that. Okay, that's supposed to be relatively informal. So proteins are enzymes. They're involved in structural components. Yeah, so this flagellum is actually this whip-like structure which moves the bacteria around. Is actually a lot of protein, and they can be used for storage. So they're structural components. They do things in the cell, like to make the cake, right? So they mix the bits together and actually make it work properly and make the end product. And they can be storage components as well. And all the information on what proteins a cell will make are in the DNA. And each cell has the same DNA, but it makes different proteins. That's why an eyeball looks like an eyeball and a liver looks like a liver. Okay? Different proteins are tasked or made in those cells, and they make those different tissue types. So, I thought it'd be fun to have a movie actually. I found this online. So, a translation, let's see if it works. Let's see how it goes. Otherwise, it's going to be boring as heck. transcription movie as well, but I'll just do the translation. So this is translation. So this is making the protein from a DNA template, if you will.
Dr. Heazlewood, actually, I've got a very quick question.
Ask away.
Stem cells, they're supposed to be like a...
Plant guy, right? Yep. When you say stem cells, I think of the bit that holds the flower.
But they're basically a cell that that sort of can replicate to any other...
Yeah, so it's basically a cell type that hasn't been told it's a liver cell yet. So once a cell sort of makes that decision to be, or certain hormones or certain components within the cellular or the tissue matrix sort of tell it you be a liver cell, it could be any type of cell it could possibly be. Because if you think of a human or even a plant at the very early stages, it's only 3 or 4 cells. You can't point to one and say that's the liver cell. So until that sort of propagates to a mass and then it starts dividing into different areas, it will actually start, suddenly there will be a trip point which will say that cell there, that's a liver cell, and that's the start of the actual liver development process.
So it depends on the proteins that are around that stem cell that will
trigger proteins can trigger it as well, and then also the proteins internally produced in. So once that trigger is formed, a certain set of proteins will then be told to be made, or not told, but will be made, and that protein population will then morph the cell into an enzyme, into a liver cell. And so in terms of how, so this is, I thought it was pretty cool. Okay. So this is actually a piece of the RNA, so it comes off the DNA. This is like the code, okay? This is the cookbook. So this is the gene that's actually been essentially taken off the DNA and it's sent out of the nucleus. You don't have to worry about that too much. So this is the template for how to make the protein. So that DNA code is essentially on that thing. Now this is a ribosome. This is actually a protein as well. And these are tRNAs. And so this thing actually jigs along and reads those different bases and sucks in a different tRNA and actually starts making the protein at the top. This is a cartoon rendition of what we think goes on. But this has been many years of work trying to sort of understand how this is working. I thought it was a pretty cool sort of video in terms of just showing the dynamics of the process. This is how you actually make a protein. So you imagine when you're trying to make a liver cell, there's hundreds of thousands of different proteins that have to be made. And so each different gene that is sent out of the nucleus, if you will, and each one has to be turned into this protein chain here. And it's this protein that then goes off and does a specific job or task. And these are basically the templates to make the protein. This is ribosome and these are tRNAs. And it's reading along the what we call messenger RNA, but effectively it's a single strand version of the DNA. You can watch this for hours, but I watch it all the time. I love it. So that's so that's how you get a protein. Okay, so they're all coded for in the DNA, and it depends on the ones you make. Will actually dictate the cell type and all sorts of functions that are occurring in the cell. So the other thing to realize is that, and in humans and in most higher plants as well, and most animals, there's probably about 30 to 40,000 genes that the DNA. Yes.
Did you say the DNA stays the same as it produces different types of proteins?
Yes, pretty much the DNA is fixed in every cell.
So how does it know?
How does it know which ones to make?
Yeah.
Well, there's a huge, it's proteins again, often, that actually do the regulatory process in terms of telling the cell which genes to actually shoot out and actually do that process of synthesizing that specific protein. Yeah, it's a really complex procedure of signaling, hormones, feedback loops, all sorts of things that are occurring. So it's not a trivial process, and we still don't really understand the details very much at all. So, so like I said, so most animals, most plants have about 30 to 40,000 genes in each cell. Okay, so they have the capacity to make 30 to 40,000 different proteins. But you've got to think in a single cell, one gene can actually, it keeps making the protein. So you can have millions of proteins in a cell, so millions of different proteins. So in one cell, of the 30,000 genes, maybe 10,000 of them get converted into proteins, but you'll have 1 million of those proteins if you counted them individually. Okay, and that's going to determine the cell type and the function of the cell. Okay, so again we're back to E. coli. So E. coli, you can imagine, is really like we tend to regard it as pretty simplistic. There's only about 3,000 proteins as compared to you guys, which have about 40,000 genes that could potentially be made, different ones. And And you can sort of just think of it like, I guess, as a single sort of bag. You've got the DNA hanging around, then a bunch of proteins which are doing all sorts of processes, like eating sugar and things like this, and replicating essentially. But so the work, and this is what Adam would have talked about, and Andrilla, when she talked about it, she would talk about microbial communities. They tend to deal in communities a bit more because they tend to be a bit more a monoculture of cell types. But one of the things we have to deal with in plants and animals, which is there's a reason I'm getting to all of this, is because that's what we have to deal with at JBay, and what I've sort of been doing for the last 10 years, is so that's sort of an equivalent. That's an E. coli, right? So it's a single bag of proteins, if you imagine. Okay, now the difference between that and an animal cell or a plant cell is this thing here is essentially the same size and has about as many proteins that get targeted there. I shouldn't have gone into that yet, but that's what I'll talk about in a second. So we have all of this compartmentalization in more complicated cells. Okay, so we have plastids, we have Golgi, we have ER, we have nuclei, we have vacuoles, pretty much the same things. We don't have plastids in animal cells. So not only do we have 10 times the number of potential genes or proteins, but we have this huge degree of complexity within the cell. So you can imagine E. coli is pretty much just a little bag of proteins in there, and an animal or plant cell is a bag full of little bags full of proteins doing different things. Yeah. So the complexity level becomes certainly far higher when you have to start to say, okay, how does the plant do something? You're actually dealing with a much more complicated structure than when you ask, how does an E. coli doing something? It's a tiny bit more simplistic or simpler to deal with. I'm not saying it's easy, but okay. So in plants, I'm a plant biologist. So in plants, we have a model plant like E. coli, sort of I guess the model microbe or yeast, or Drosophila, the fruit fly. Anyway, so ours is called Arabidopsis, and it looks like that. It's a weed, basically. This is the publication of its genome. So the genome was sequenced in 2000, which was a huge breakthrough, obviously. That was sort of laid down the foundation of what we call functional genomics work. So sort of once you have a genome, you can actually start to sort of understand what's going on at the genomic level. You can actually count how many proteins a plant could actually make, or this particular plant. And there's all sorts of different sort of tricks that you can use once you sort of have a genome. And so this was a huge breakthrough in terms of plant sciences. And that's pretty much, if you come down to JBay, that's pretty much what we use to do our cell wall research. We do a bit of a rice program as well, so we use rice. It's a good model as well, but they do that up at Davis. But you'll see Arabidopsis, you'll see a ton of them down there, and they sort of grow to about this big. So once you have the genome, I guess like I was saying, you can actually infer the number of proteins. So once you sequence this genome, and JGI, obviously part of the LDL, is sequencing genomes, and I think they're even part of sequencing some of the Arabidopsis. They're certainly part of sequencing the human genome. You can basically use computers, because we've worked out we've broken down the code. That was Watson and Crick. They won Nobel Prizes for that. And so we can just use a computer, and we can sort of scan along the DNA and actually infer how many proteins a particular Arabidopsis can actually code for. And we know that Arabidopsis codes for about 30,000 proteins. Okay, so I've talked to you about the complexity of the cell, and here's where I talk about it's a bag full of little bags of proteins. So if E. coli only has 3,000 proteins, why does a plant have 30,000? So essentially what's happening is each of these little bags contains a different population of proteins. Because each of these little bags, these little areas, these organelles, do a different thing in the plant. And together, they make the plant cell a plant cell, and they do what the plant actually does and turn into a plant. So you can see that we have a high order of complexity. And the way, we tend to use this analogy of an address, a postmarked envelope. How does it get to, what is that, Georgia? I've got no idea. How does it get to Georgia, right? It has an address on it. And it's the same thing with proteins. So even though the genome has 30,000 genes, they can convert those into proteins, they have sort of basically an address on them, which will send them to a specific location to do their function. Yeah? Good. So, Arabidopsis codes for 30,000 proteins. And the problem is, most of them, we have no idea what these proteins are doing. Okay? Absolutely none. It's been 10 years now, and various other sort of piecemeal work that was done in the 80s and 90s. But really, we don't have a clue for about 70 or 80% percent of the proteins, we have no idea. We have some sort of functional idea or some idea of what they may do, but we have no actual detailed idea of what they're actually doing in the cell. Okay, so those little bags are subcellular organelles. So each one tends to have a major function. So you saw photosynthesis, the green one here, the plastid. That's where the light is converted into sugar, basically. Okay, so during photosynthesis. So we know a huge emission, it's a DOE emission of this little organelle here, is to undertake photosynthesis. So we can sort of start to guess, if you know where a protein is in the cell, you can start to infer some functional interaction. So you can say, if it's in the plastid, it probably has some relationship with photosynthesis. It's certainly going to be somewhat clued into that sort of process. So we can start to infer function once we know localization. The other thing is proteins tend to work together. Yeah? So once they're in the same little bag in the cell, there's a good chance they'll interact. So again, we can infer functionality. And then we can start to say, we contribute functionality to the unknowns, because most of them we don't have a clue what they do. So it's actually quite useful to understand where proteins go in a cell. So here's like an illustration of them working together. So this is a lipid membrane. This is actually in the membrane of mitochondria. That's what the mitochondria looks like. This is like the outside the bag bit of the mitochondria. And these are proteins sitting within the membrane. And there's about 78 or 80 proteins all in here together, working together. And essentially what they're doing is converting sugar to energy. Yeah? So you have mitochondria and it's making ATP, which is sort of like your battery in your body, which goes off and can do various energy processes. But it's just to illustrate that once you co-localize unknown proteins into a mitochondria, for example, you can start to infer its processes with regard to respiration and basically energy production. So it's really quite useful. So how do we put plants? Because most people tend to think, well, plants are just green and they sit there and they don't do anything. But if you do it in terms of what's our current sort of knowledge about, so all of the model systems, Drosophila, yeast, zebrafish, mouse, human, they all have this same issue of we don't really know what all the proteins are doing. And if you put it in terms of a score, plants actually do pretty well. So humans do a bit better, mouse does the best of all. And this is basically number of gene products that we know biological, functional, subcellular component with. Okay, that's what these are the lines for. And that's sort of telling us how many genes can we sort of allocate to some sort of tenuous function. And so in terms of the model systems, plants actually do pretty well. We've actually done quite well, but we've still got 3,000 or 4,000 proteins that we have some information about. So you can see we're miles away from really knowing anything. And that's the same for all of these model systems. Okay, so you heard a bit about mass spectrometry or proteomics, the term before. So one of the things that I've sort of been playing around with for 10 years now is basically trying to pull the cell into its little parts. So the mitochondria where the battery is made, the ER where protein synthesis occurs, the plastid where photosynthesis occurs. If you can basically pull those out of the cell, we have a technique which is about 10 years old now called mass spectrometry or proteomic-based mass spectrometry, where we can take these sort of bags of protein, we can throw them into the mass spec and we can actually identify the proteins that are in those bags. And we reference that back to the genome, right? So it's an informatics or a computational process. And so two areas, and certainly what I'll talk about today is the Golgi apparatus. Okay, so the Golgi is actually really important in cell wall biosynthesis. I'll talk about why we're interested in the cell wall shortly. So that's a subcellular compartment within the plant cell. It's involved in synthesizing the cell wall, which we're interested about at JBay. But really we didn't know really what the proteins that were in that little bag were. So the idea was to basically isolate this Golgi and look at the proteins. What are the proteins that are actually in there doing the functional work to make the plant cell wall? Another video, excellent. Should have had more Mickey Mouse videos. Okay, so mass spectrometry. Again, this was really cool. I had to find it. It's actually a company. So how do you get your sample into the mass spec? So you basically grind up the sample and you spray it into the mass spec. And what a mass spec is an instrument that can measure at a really high accurate level the mass of a molecule. Okay, in this case, a protein. So this is it's a TOF, time of flight, and essentially the mass analysis component. So here we go. You spray in your sample nebulizer. Yeah, here we go. So you basically spray in your sample in a droplet, and it ionizes. Yeah, so this would be your protein. And it goes into the mass spec through little ion guides, through ion capillaries. Just gets rid of junk, basically. So these are the proteins going in here. And the whole idea is you want to measure the mass of the protein. Because if we can measure the mass of the protein accurately to 4 or 5 or 6 decimal places, we can actually use computational methods to determine what that protein is. Okay, and here's the pulsar. So it's a time of flight. So basically, literally, it's time of flight. The time it takes for the protein hits the mirror and back down to the detector. The bigger proteins will take longer, the smaller proteins take a shorter time. And from the time it takes to fly through that chamber, it can actually work out the mass. Here it is hitting the photon scintillator, yep. And so basically that will get converted into a signal. So it's basically measuring mass. So this would be masses along here. So 2,000 mass units, 3,000 mass units, 4,000 mass units. And so when you put in a complex sample like that, you can actually determine all of these different masses, which you can then use to work out what this red line actually is in terms of the protein. It's protein X, or protein 57 from your informatics. And so that's the sort of output you get. So you get a bunch of masses from the mass spec. So you've isolated a subcellular compartment, you've put it into the mass spec, and you get different masses. And then you basically use a computer. We have the genome. We know we can theoretically assign the masses of all the proteins. So we can run through, predict all the proteins, and then we can just use, we know all of the masses of all the elements. We can just work out the masses of the protein, yes?
What if a different type of protein was found that was the same weight as a similar protein you mistaken it?
Yep, that's one of the problems. So there's actually a separation component in front of that. It's a good question, actually. So you actually add the sample on and you'll do a separation, basically an affinity separation.
And how do you, what other variables do you look at besides weight to differentiate them?
Well, that would be retention time in that regard. So, this LC, this separation process, retains proteins differentially. So ones that are exactly the same mass will have different properties, so they tend to get retained differently. So they'll hit the mass spec at a different time. So there's that other dimension. So we have the mass dimension, and we have the time retention. But there are always complexities with this problem. We always do get overlaps. So it's certainly a messy, you can see it's quite a messy business, right? You basically get rid of all the junk there and you just keep the major peaks. And so what the junk is, well, we don't really know, so we just get rid of it. But that's computational work. So what I'm trying to show here is you have a computational list of proteins and you'll have your experimental masses up here. And you can basically say, I saw that mass there. So that protein was in my sample, in my bag that I isolated out of the cell. Yeah? So that's how we do proteomics matches. Or that's how we're doing proteomic characterization of these different subcellular compartments to try to work out what proteins are there and doing a function. Okay, so JBay, why are we even bothering to do this? Why are we interested in the Golgi apparatus, which is that funny substructure that I was talking about, which is involved in plant cell wall biosynthesis? So the objective of JBay is to basically use plants, so they basically just grow. Once you have gardens, I imagine, you put plants out there, they grow, they produce biomass. You know? And it's this biomass that is what we're interested in, because that is essentially sugar. It's essentially complex sugar. It's about 70% sugar. I'll show you a breakdown of that shortly. And so, the problem is it's pretty hard to break down that. If you eat straw out of the field, you're not going to get much nutrients coming back with it. Even though it's essentially sugar, you're not going to get anything. And that's because you have to have a big cow with 4 stomachs to actually break it down. Okay? So we have to use like a mechanical pre-treatment process, and that's what Blake Simmons works on, who's from San Diego National Labs, but also working at JBay. There's a whole team that works at trying to break this biomass down, and then you digest them up into monomers, into like sugar molecules with enzymes, and then that gets fed to microbes, which is what Jay Kiesling's doing, which is engineering of microbes to make a fuel. And so the idea is that we think we can sustainably grow a sugar supply, which is actually hard to extract the sugar out of, but if we can then feed it and make a biofuel. And that's the objectives of JBay. So really it's to sort of replace this paradigm where we're drilling and refining and making petrol for gas for cars, we call it petrol. And actually use lignocellulose biomass, which is renewable. We can just keep growing it relatively easily, refine it out in the field, and have a drop in.
And so what are the sources for the biomass?
What are the types? What are the types of plants?
Pardon me, so what are you using for the being successful? That isn't like a food crop?
So we're actually currently sequencing the genome of a plant called switchgrass. And so that, and like I said, we need the genome because to do all of the functional genomics work, the manipulation, we really do need a genome. So so we use switchgrass, which actually that's it there. It creates a huge amount of biomass. It's perennial, so you don't pull the plant out from the roots. You actually just crop it back, and it'll reshoot again.
Are you looking at other things as well? I thought I heard eucalyptus.
Miscanthus? Oh, eucalyptus. Well, it depends on where you are. I mean, the Midwest, because they grow, it really is a crop type environment or agricultural environment. That's the sort of plant you want to grow. But if your area is a wooded forest or something like this, then for sure you could use eucalypt or pine, anything. That's all of it is high amounts of sugars, which is what I'll show you here. So imagine even a tree or that grass, the switchgrass I was showing you there. So the cell wall, which is the hard bit, basically. If you think of straw, that's all just cell wall. Okay, so that brown straw that you were seeing there. And what it is like a big matrix full of sugar molecules that are all interlinked and linked together. You can imagine it like reinforced steel. We have members of steel, which is actually not a sugar polymer, but it's a component of the cell wall. And if you can imagine, cement is basically the sugar, all interlinked and interlocked together. And there's a huge amount of this sugar in cell walls. It's just really hard to get it out. And it's nearly 70, in the order of 70% sugar is a plant cell wall. And we have this other proportion of lignin, which we actually don't want. And that's part of the deconstruction technologies to actually remove that and retain the sugar types. And so you notice that this hemicellulose, there's two sort of components. One's called hemicellulose, which is essentially glucose, honey or something like this, or sugar that you put in your coffee, and hemicellulose, which is a mixed sugar population. And it's the hemicellulose proportion that is made in the Golgi. Here it is here. So this is a cartoon of the Golgi. This is a plant cell here. This is the sort of the cytosol, the juice that it sort of floats in the cell. And this is the Golgi here. And the Golgi makes that hemicellulose contents of it. Yeah, hemicellulose. And that's an example of a hemicellulose. These are sugar molecules joined together. You can see they're sort of branched off, crazy little sugar molecules all together. And that all gets made by thousands of proteins in the Golgi apparatus. And so what we'd like to do is be able to manipulate this sugar. So one of the problems is that sugar, that cell wall material, that mass, is not particularly valuable. It's very light, so it's not very dense. It costs a lot to truck it around. So if you can change, and the profile is not ideal. We have sugar types here like xyloses and rhamnoses and things like this. Not glucose. We'd love glucose, would be the ultimate. But we have some other funny sugar types. So if we could understand what's going on in this bag here, this Golgi, all the proteins that are working together, and how they make these sort of structures, we can potentially manipulate how this is made and actually reduce the number, say, of these light purple ones, okay, which are not the best sugar type. And that would actually, and maybe replace it with one of these yellow ones, which is a good sugar type. And that would actually increase the value of the plant stock out in the field. And that would actually, even saving percents like that, downstream has a huge impact in fuel production. Okay, so onto actual science. So this is actually, I actually work with, so this is Arabidopsis in a cell culture. So you can actually put a plant and grow it in a liquid culture, shaking around. And so the idea was to use, so various people have tried to isolate that Golgi, that thing that's making the matrix polysaccharide content, out of plants before. But one of the problems they've had is that the classic method was not very good at getting purified Golgi bodies out. So we had to use a new instrument called a free flow electrophoresis, which we bought when we arrived at JBay. So the idea was to purify these away from all the other components and put them into the mass spec, like I showed you before. So we could work out what are the proteins that are actually in here that are making the matrix polysaccharides, which are going into the cell wall. Okay, so here's one of the problems, of course, and why we had to use what we call the orthogonal approach, or this free flow electrophoresis, is when we do, and basically this is like, you basically take the plant, you grind it up in buffers and liquid, and you spin it in centrifuges with sucrose gradients and all sorts of stuff. And this is what you can't see that thing, right? All you see is grey juice, right? And this is what you imagine. You say, yep, it's full of Golgi, right? That's fantastic, that's what we want. But really, that's what it looks like. You get a bit of plastic in there. So you can enrich for these guys, but you can never, it's really difficult to remove all these other contaminating organelles or little bags or other bags of proteins. So you imagine if you throw that onto the mass spec, it's really hard to say, well, what's Golgi and what's plastid, right? It's really difficult. You want this to be as clean as possible so you can say, these are the proteins I found these are what's in the Golgi, these are making matrix polysaccharides. This is what I want to understand functionally so I can manipulate matrix polysaccharide biosynthesis. These contaminants are going to mask your interpretation. And there's all sorts of reasons that's happening. I mean, basically, these things are not just floating little free bags in the cell, right? They're actually interlinked and they communicate with each other. They're locked in through protein complexes and what we call cytoskeleton, which basically locks the organelles together. So it's actually really hard to do this. This is usually the normality. And so what we had to do was, like I said before, use this thing called the free flow electrophoresis. And here's the classic way. Here's actually what it sort of looks like. This is a Golgi enriched fraction here, okay? So that's all we're dealing with. We don't see little bags, little purple bags. We see little grey band, basically. But that band there basically looks, the constituents are like that. We know that because we put it on the mass spec and had a look to see what it looked like, and it was full of stuff from all over the place. And so this was the traditional way you would enrich the Golgi out of the plant, or an atom cell for that matter. And so the step that we took to try to really see whether we could clean that up was to use free flow electrophoresis. Now, this is what we would call a density centrifugation. So basically, sugar is pretty dense. Golgi have a certain density. Other organelles have different densities, and so you can actually spin with an ultracentrifuge. I don't know if you've seen the centrifuges. You spin them really, really fast. You can actually float a different population at a different density up through this sucrose, which is what we've done here. And this has been done for 30, 40, 50 years. But it was the addition of taking that enriched fraction and putting it on a free flow electrophoresis device. Now, what the heck is this thing, right? It looks like this, and what I'm describing here is the plate that runs sort of down here. It's about 15 centimeters across. I don't know inches, okay, so where I come from, it's metric, so that's what you're going to have to live with. It's 50 centimeters long and about 0.5 millimeter thick. You know? So basically, buffer flows through, liquid basically flows through here continually. And what we do is we apply a charge across this way. So this is the flow of buffer continually flowing through here. You put your sample in, you load it in directly on sort of on top in here, and it runs in through here. Now, the idea is that things have different densities. Things also have different charges. So if you apply a charge across this area, different organelles, like the Golgi, like the plastid, will start to run away from each other because they have a different charge. So one will be much happier to go to the positive, and one will go to the positive more slowly. So the idea is you have two separation techniques, density and charge. The idea is to hopefully clean up the sample. And that's basically what it looks like. It's actually a mitochondrial sample. So here's the flow of buffer going that way into this, and there's the electric field. And you can see it's actually moved into two different streams. Yeah? So that's the sample that's come in down here, and you've actually pulled two streams away from each other. And so at the other end here, you collect 96 samples. So this plane sort of is convoluted round with tubes and drops into 96 samples. So you get this huge fractionation capacity. You can really separate all of those different streams into a different fraction pool. Okay, so this is complicated maybe. Okay, so this is, if you can imagine, the blue line here is basically showing, so it's a 96-well plate. You have to think of it, it's basically sequentially 1 to 96, and it's sort of in these plates here. The blue line is showing the amount of protein coming off. Yeah, okay, so, and the red line here is actually Golgi protein. So what we did is we basically took fractions, each individual fraction, put them into the mass spec, and identified the proteins that were there. Yeah? So we know certain proteins that are thought to be in the Golgi. And so when we had a look at the type of proteins that we're finding in each fraction, we could actually see that the Golgi was actually coming out sort of in this slope here. And you can see for mitochondria, which I talked about before, that's an organelle that makes little energy supplies, ATP. You can see it sort of running down the bottom. It actually runs up here like this, but we haven't drawn it in. ER, which is another organelle, it runs differentially. So normally, these would all run together. I've got some more to show you. Yeah. So here we go. You see we've pulled away some of vacuole, which is another subcellular compartment, and some plasmamembrane. And the orange is now the Golgi, unfortunately. But so normally those would all be in that sample. But what we can do now is take fraction 21, which would be a really enriched Golgi fraction, and it's removed all of the other crud away. And that's what we did. So what does it actually look like? This is actually like real. So this was done by Manfred, which also part of PBD. This is electron microscopy. So it's a microscope that can go really, really close, and you can actually see subcellular compartments. So that's the whole cell. That's what a Golgi looks like. It's like this is a cross section through that sort of stack. It's called a Golgi stack. And you can see as we disrupt the sample, it starts to fall apart a bit. And then as we go to that sample that was prior to putting it onto the FFE, you can see there's probably, it's basically a mixed membrane. So if you analyze that in the mass spec, I mean, you just see a whole lot of junk, basically. And you can see, after using the FFE, we're actually getting a very distinct membrane or population of vesicles, which seem to be purified Golgi, because that's the fraction that had that really high amount of Golgi. So we're actually pretty pleased what was going on. And so we threw those samples into the mass spec, right? And we could identify the types of proteins. We could give them a functional name based on similarity to other proteins in the system. But of course, we found it's probably underestimated. It's probably about 20% really that we have no idea what they do. There's a bunch of contaminants still, so we still have components from other cell compartments. But what was really nice to find was that 20% of what we found in that protein, we found about 500 proteins, were involved in matrix polysaccharide biosynthesis. So these are the enzymes directly involved in making the cell wall. So that was really useful because now we have a whole cohort of enzymes that we can start to look at manipulating or understanding how they work as targets for manipulation. We found various other components. Okay, and so that's just a cartoon of the sort of components of things that are going on. So we have these things, glycosyl transferases, which are actually making the matrix polysaccharide. But there's a huge amount of proteins that are all interrelated, all sort of doing jobs to enable sort of these GTs, these glycosyl transferases to work. Bringing substrates in, converting substrates, methylating, and all sorts of things. So what it really tells you is this little bag of proteins is hugely complicated, and making matrix polysaccharides is also an incredibly complicated process, which I think we always knew, but this was sort of even more confirming it. Okay, so what did we actually find? So did we actually find anything of interest at all? Has it moved science forward, I guess, other than here's a list of proteins? So I've talked about these GTs, so these glycosyl transferases are proteins, okay? So it's an enzyme that's making a matrix polysaccharide. This is supposed to be a chain of polysaccharides. Okay? And they actually use sugar groups that come in from the cytosol, the juice, okay? So they get transported into the Golgi. This is the blue thing is the Golgi. And then sugar gets put onto this growing chain. So those are chains of those sugars which are going to the cell wall. And so one of the carrier components is NDP. It doesn't really matter what it is. It's basically a carrier of the sugar. It gets released once this sugar gets attached. And then there's another enzyme which people have known existed in the Golgi, but they've never actually been able to say, oh, that's the gene that makes this protein which converts NDP to NMP, basically releasing a phosphate. And this is really important because this drives the whole reaction here. Okay, without this occurring, this builds up and prevents this from occurring. So what have we known about these things? So we found some candidates that could have actually done this enzymatic process, this NDPase reaction, so removing the phosphate group from NDP. But in the literature, for 10 years, these two genes have been reported to be on the plasma membrane, so out here. So someone had mislocated them, but we found them in our Golgi. We were thinking, okay, maybe it's contaminants we have. So there's the Golgi there, there's the plasma membrane. So what we did is we can use a little trick called Fluorescent Tag. So we basically take one of these, so we have the proteome, we're pretty sure it's there, but other people have said these genes are in the plasma membrane, so a different part of the cell. So they'll be doing something functionally completely different, not involved with matrix polysaccharide biosynthesis. But what we can do is use a fluorescent tag on this gene that we found, this protein that we found, this NDPase. We can use another gene, so we put a yellow tag on it. We basically use a tag that we know goes to the Golgi. So these are actually Golgi here, these are little spots. And we put a blue tag on that. And then when you put those into the cell together and then do an overlay with a confocal microscope, if they co-localize, the color will change because they're in exactly the same spot. So we actually proved using a fluorescent tag, so it proved our proteomics was good, isolation was good, and it proved that this thing that had been thought to be on the plasma membrane was actually in the Golgi apparatus, where we hoped. And then to actually really prove that it was doing what we thought it was doing, it's more complicated, but I thought let's get into some science. So there's if you take yeast is a classic model for doing what we call functional complementation. So you can prove the function of a gene or a protein by putting it into yeast. And if you look at this first one here, so this GDA1 is actually, so this is a wild type yeast, this is a GDA1 mutant yeast. And this mutant yeast doesn't have that NDPase. Okay, so the one we just identified in the Golgi, it it doesn't have it. The yeast grows fine, but what it does do is produces a shift in protein glycosylation. So protein glycosylation are sugar groups attached to the protein. It's the same process as making matrix polysaccharide. So you just don't get sugar groups on some proteins. And so you get this band shift. This is basically a size differential as you run a protein through a gel. And what we could do is take the Arabidopsis, so the plant protein, put it into this yeast background, forget about these two, and we could recapitulate the wild type phenotype. So this is the yeast mutant, which should have a band shift. We can actually bring the band up. So that means our enzyme can work in yeast and can attach sugar groups to a protein. Okay? Which means we've functionally proved that that protein is an NDPase because it complements the yeast version. It's located in the Golgi, and it was thought to be in the plasma membrane. So that's sort of the power of sort of trying to find some of these sorts of doing the whole proteomics thing. It allows you to actually understand functions and things that are occurring where there might have been completely no knowledge, lack of knowledge, or mistaken knowledge in the community. Okay, so we've published it. We've actually published this Golgi proteome now. And one of the things several years ago we found was that various people were doing various proteomes, and it would be actually instructive to know where are all the proteins, right? There's no point in, when you publish these things, you basically send them an Excel spreadsheet full of all your proteins that you found, okay? And they can't search them. They're sitting on a website somewhere. Someone else will do a peroxisome, and their list is sitting in a website as well. So what we built a couple of years ago was a subcellular database for Arabidopsis. Yeah? So basically, we went through the literature, we found all of these different studies, and we put all of the proteins together into a database online. And so, because it's really useless to the community to have them sitting locked in papers. And so there's all sorts of various mass spec studies. You can actually do prediction. You can predict the localization of the protein by its sequence. So we ran predictors, and there's these fluorescent protein localizations as well. And we're able to bring all of that information together. And what you can then do from a database like this is say, okay, so you can say, how many times has someone done a mitochondrial proteome and someone also done a plasma membrane proteome? What's the overlap? So these intersections here, this is the intersection of all the plasma membrane proteins that have been called, or we can talk about Golgi maybe as well. So Golgi, 88 proteins in the Golgi. And these are other studies from the nucleus. They've found 1 of that 88. These guys, these vacuola guys, they found 13 of the 88. So it really starts to allow you to assess what are contaminants, what are potentially dual located in 2 spaces. That's also something I didn't talk about. But also just to get a feel for what are the major contaminating membranes in these systems when we're all trying to work out where all these proteins are.
Why are there different types of proteins why are there Golgi proteins in like a mitochondria or something?
So more than likely, it would tell us that it's a contaminant. The classic mitochondria and chloroplasts, they're really hard to separate from each other. So the overlap between the 2 of them is really bad. So nearly a fifth of the things being called in the mitochondria are also being called in the chloroplast. So it basically is a technical limitation, that purification, those 2 there. So it allows you to start to assess those sorts of things. So until we built that, you could never really assess what the hell the story was. So people were claiming, oh yeah, my protein's in the chloroplast. Well, it's probably actually in the mitochondria, to some degree. So one of the other things, so this was a database, and like 10 years of Arabidopsis proteomics, we've had various databases. So super, actually, we built that when I was in Australia, this subcellular database. But there's been a ton of databases that have been developed all over the world by different people doing different sort of proteomic studies. Yeah. And so when I came here, it was actually really annoying when you want to know information about a protein that have been worked on with proteomics. You either have to visit all of those databases to get the information on a particular protein. You're interested in one protein, you've got to visit all of these places to find the specific information that you wanted to learn about. Right? And it was pretty annoying, actually. So I actually contacted all of my colleagues who've been working. So most of the plant proteomics guys, most of us know each other. And these are all the different databases. They all sit in Switzerland, in Australia, in Germany. And what we decided to do was create an aggregation portal, right? So where we actually had one site where you could go and visit, and there it is there, it actually lives at JBay. You could put in your protein code, if you will, like the identification code for the protein, and you would get proteomics information for it. This is a bit of an aside, but this was just something that you can see in terms of when you're doing this functional genomics that sort of occurred, the amount of data we're all producing is extraordinary, and we're sort of losing track of it. I don't know if Adam talked about the K-base, but that's sort of part of this whole concept of trying to now bring all of this data together.
So is it only limited to JBay, or
No,
is it shared?
it's housed at JBay. So we run it at JBay, but it's for the international community.
So it's shared?
Yeah.
And it's an open source?
Yeah. So, okay, so I guess that some of that stuff was slightly off topic, I guess. But anyway, I guess the summary of this is, where are all the proteins? Well, we still don't actually know where the hell the proteins are, right? So we only know about 5,000 of the 30,000. And do we care? Well, yes, because it can actually inform us about function. It also can try to clear up issues, like I showed you with the NDPAs. And this sort of knowledge can help drive, manipulation and understanding of plant systems, which is really what we're trying to do at JBay.